Welcome to This Explainer. Let's just dive right in, shall we? You probably already know that every single living organism out there, including you, carries a vital biological blueprint. But really take a second to consider the sheer scale of it all. Your DNA contains over 3 billion base pairs, and they're packed tightly into almost every single cell in your body. It's basically this instruction manual of unimaginable complexity, and it governs absolutely everything about how biology actually functions. But here's the thing. Knowing that blueprint exists, that's really only half the battle. The actual real challenge is figuring out how we read it. I mean, how exactly do we take this microscopic, super tightly coiled molecule and actually extract actionable data from it? Well, that is exactly what we're going to be breaking down today. Okay, so let's start with section 1, the genetic code, the foundations of life. At its core, DNA sequencing is basically the fundamental process of determining the exact sequential order of our biological alphabet. Think about it like this. When scientists are sequencing DNA, they aren't just, looking at a random molecule. They are literally reverse engineering the biochemical code that built you. They're reading it base by base to understand the exact program that's running just underneath the surface. Now, as you probably already know, that entire genetic alphabet consists of just 4 letters, or bases. A, T, C, and G. But the true marvel here is the absolute data density. The exact arrangement of just these 4 simple variables actually dictates the difference between, say, a blade of grass, a bluebird, and a human being. It's honestly the ultimate example of super complex data emerging from really simple, elegant rules. So why even go through the massive effort to read this code in the first place? Well, because decoding it basically gives us the ultimate biological cheat sheet. We aren't just looking at genes for the fun of it. We're hunting down the precise mutations that drive complex diseases. We're tracking viral evolutions in real time, and we're even mapping out the entire evolutionary tree of life. It is hands down the definitive tool for modern biological discovery. Which brings us to section 2, how sequencing works, the process pipeline. Now, the physical pipeline for this is an absolute masterclass in modern automation. First things first, the DNA has to be collected from a physical sample. Then it gets loaded up into these high-throughput sequencing machines, using techniques like next-generation sequencing, which chemically read literally billions of fragments all at the exact same time. And finally, that physical organic chemistry is instantly converted into digital raw data. This massive parallelization is a real game changer. It allows us to process biological info at a speed and scale that was honestly totally unthinkable just a few decades ago. All right, let's move right along to section 3, making sense of data, the translation. So generating all this digital data is kind of like scanning a massive book that's written in a totally foreign language. I mean, just a single human genome can generate hundreds of gigabytes of overwhelming raw data. To bridge that gap, going from raw data to actual meaningful biological insights, we have to rely on a field called bioinformatics. Bioinformatics essentially acts as our translator. It uses massive computational power and really complex algorithms to interpret that total avalanche of information, digging in to find those critical, meaningful signals hidden right there in a vast digital haystack. And finally, that brings us to section 4, real-world impact, expanding genomics. This translated data, it is literally the engine driving modern medicine today. It's allowing oncologists to pinpoint the exact molecular drivers of a specific patient's tumor. It helps epidemiologists track infectious diseases as they mutate on a global scale. And, perhaps most excitingly, it's the absolute bedrock of personalized medicine. It's moving us away from those old one-size-fits-all treatments and toward therapies that are built specifically for your own unique genetic code. Decoding the human genome was without a doubt one of science's greatest achievements. But actually, scratch that, it wasn't the finish line, it was really just the starting line. Today, DNA sequencing forms the unshakable foundation for the entire field of genomics. It is actively taking things that used to sound like total science fiction and turning them into standard medical practice. It's setting the stage for major breakthroughs that are for sure going to completely reshape global healthcare. Which leaves us with a pretty thrilling question to ponder. We've crossed this massive threshold in human history, right? We've finally learned how to actually read the blueprint of life. So, what exactly are we going to build next? It is a wild journey into the ultimate potential of our own biology. Well, thanks so much for joining me for this explainer, and I hope you found it as fascinating as I do.